In blue water without shelter, a glass-like shrimp drifts while sunlight passes through most of its body. Its outline nearly vanishes, leaving only eyes, digestive tissues, and small reflections suspended in the open sea. Here, concealment cannot depend on rocks, vegetation, or a dark seafloor. The animal hides by reducing the optical difference between itself and the water behind it. Transparency seems simple, yet living tissue contains membranes, proteins, organelles, pigments, and fluids that scatter light. Every surface can betray the body when illumination arrives from changing directions. The mystery is how an animal remains functional while making those internal boundaries difficult to detect. Open water predators search for contrast, motion, polarization, silhouettes, and flashes rather than named anatomical parts. A transparent body must defeat enough of those channels at useful distances, not achieve perfect invisibility. The strategy works best in small, watery animals whose tissues can transmit light with limited scattering. It becomes harder around dense muscle, mineralized structures, food, reproductive organs, and light-sensitive eyes. Evolution, therefore, conceals some components while tolerating or actively disguising others. Our investigation follows a beam of daylight through the animal, then asks what still returns to a predator. Each optical solution carries costs for movement, sensing, digestion, protection, or reproduction. The drifting shrimp survives inside those compromises, transparent from some angles and conspicuous from others. Light entering seawater is absorbed and scattered, with red wavelengths disappearing faster than blue. In the upper ocean, downwelling light creates a bright field above and dimmer directions below. A visible object differs from its background when it absorbs, reflects, refracts, or scatters incoming photons. Transparency reduces absorption and scattering by matching refractive properties across adjacent tissues as closely as biology permits. Water-rich cells and thin structures can create fewer strong optical boundaries than thick, complex organs. Gelatinous zooplankton exploit this principle. Because much of their volume is seawater held in delicate tissue, salps, ctenophores, hydromedusae, and larvaceans include species whose bodies are substantially transparent. The condition is not emptiness, because muscles, nerves, digestive systems, and reproductive tissues still operate. When you see through a salp, light has crossed organized living material rather than a hollow bag. Small size also shortens the path through tissue, reducing opportunities for absorption and scattering. This helps explain why transparency is widespread among larvae and planktonic stages. As bodies thicken, even weak scattering accumulates, and complete optical transmission becomes more difficult. Some animals remain flattened or ribbon-like, minimizing thickness along common viewing directions. Others distribute muscle into thin sheets instead of bulky, opaque masses. The cue is broad daylight. The animal maintains a low-contrast posture, and distant visual predators receive a weaker target. Posture matters because a transparent edge can disappear, while the same body viewed lengthwise reveals more tissue. Behavior, therefore, complements anatomy, adjusting orientation to the prevailing light field. Experiments using controlled illumination can measure contrast from different angles, though tanks simplify natural backgrounds. Field cameras reveal appearance in context, but camera sensors do not exactly reproduce predator vision. Optical measurements quantify transmission, while anatomy identifies the structures, causing residual reflections. Together, these methods test concealment without claiming to experience another animal's perception directly. Eyes present a stubborn problem because vision requires pigments that absorb light. A completely transparent retina would fail to capture the photons needed for sight. Many transparent animals retain dark eyes, creating two conspicuous points within an otherwise faint body. Some reduce this signature with small eyes, reflective layers, elongated shapes, or surrounding transparent structures. Hyperiid amphipods include remarkable optical arrangements that distribute retinal material across enlarged eyes. These designs gather light while altering the concentrated silhouette a predator might detect. The functional interpretation is supported by anatomy and optics, but the exact survival advantage is difficult to measure. Digestive organs create another problem because captured food may be colored or opaque. Some transparent predators have narrow guts that minimize visible area. Others position digestive tissues along body axes where silhouettes are already unavoidable. The cue of prey contact triggers capture. Digestion concentrates material, and the consequence is temporary increased visibility. Faster processing or behavioral retreat may shorten that vulnerable period, although direct demonstrations vary among species. You should not imagine transparency as a permanent cloak independent of feeding state. Reproduction can likewise reveal animals when gonads fill with eggs, pigments, or nutrient-rich tissue. Seasonal changes may turn a nearly invisible juvenile into a more visible reproductive adult. Selection balances current concealment against the need to build viable offspring. Some crustaceans conceal reflectors by using irregular microscopic structures that reduce directional flashes. Ordered surfaces can act like mirrors, producing bright glints when their angle matches the light. Disordered arrangements spread reflection across directions and wavelengths, lowering a sharp signal. Research on transparent shrimp has shown cellular organization capable of suppressing conspicuous reflections. Such evidence links microscopic structure to measured optical performance, though predator responses require separate testing. Internal fluids also require refractive matching because mismatched interfaces bend and reflect light. The closer tissues approach the refractive index of seawater, the weaker many boundaries become. Exact matching is impossible across all wavelengths and structures, especially in metabolically active organs. Transparency is consequently graded wavelength dependent and sensitive to viewing geometry. Damage can make transparent tissue cloudy when disrupted surfaces scatter light more strongly. Stress, disease, or development may likewise change optical properties in living animals. Bioluminescence sometimes accompanies transparent bodies, but producing light serves different functions from transmitting ambient light. Counterillumination uses downward light to reduce a silhouette against the brighter surface. Some midwater animals combine transparent tissues with photophores switching strategies as daylight fades with depth. Q is the overhead light field. Photophores adjust output, and the consequence is reduced contrast from below. Evidence for counterillumination includes anatomy, light measurements, and observed matching responses in several taxa. It remains distinct from transparency because emitted photons replace a shadow rather than pass through tissue. At greater depths, daylight weakens until black or red pigmentation can become effective concealment. This creates a vertical transition among transparency, counterillumination, silvering, and dark coloration. No boundary is absolute, since depth, time, water clarity, and predator senses continuously change. If you descend beside the shrimp, its vanishing act would weaken as the surrounding light field changes. Open water ecology rewards concealment because attack can come from almost any direction. Transparent prey reduce encounter distance, potentially gaining moments to escape after detection. Transparent predators can also approach prey without presenting a strong, advancing silhouette. The same adaptation, therefore, shapes both pursuit and avoidance within planktonic food webs. Environmental change can modify its effectiveness by changing water clarity and spectral light. Sediment, plankton, blooms, and dissolved organic matter alter background brightness and color. Warming can shift species ranges, exposing transparent animals to different predators and optical conditions. Plastic particles or oil droplets may enter food webs, but their effect on transparency varies and remains insufficiently known. Artificial light near coasts can create unfamiliar directions and intensities, possibly revealing animals adapted to natural cycles. These pressures matter because concealment is calibrated to an environment, not manufactured as universal invisibility. Future interpretations must reflect measured predator sensitivity rather than human photographs alone. Field experiments can compare attack rates while controlling motion, size, and brightness. High-speed imaging may reveal whether animals change posture immediately after detecting shadows or pressure disturbances. Repeated optical measurements through development can show when tissues become transparent and which organs remain dominant. Measurements across feeding states could quantify how captured prey temporarily changes the animal's detectable contrast. Researchers should measure living animals because preservation often clouds tissues or changes their dimensions. They must also report angle and spectrum, since 1% cannot summarize transparency in every condition. You can return now to the beam, crossing the glass-like shrimp near the surface. Most photons continue onward, while a smaller fraction exposes eyes, gut, edges, and movement. Predators may integrate those fragments, especially at close range, or against an uneven background. The animal survives not by erasing itself, but by delaying recognition until escape becomes possible. Comparisons among relatives can test whether thinner bodies, refractive matching, or behavior contributes most. Candidate structural proteins matter only when their presence corresponds with measured optical effects in living tissue. Direct predation observations will show whether laboratory contrast predicts survival in turbulent natural water. Unknowns will remain because every observer, camera, fish, and jellyfish samples light differently. The opening shrimp will never be invisible from every direction, wavelength, distance, and moment. Its scientifically defensible achievement is narrower and more elegant. Living tissue makes itself sufficiently similar to illuminated seawater to escape notice.